single unit, we call as dimer. If it is joined with three, we call as trimer. If it is called as if it joined with those are called as oligomers. If more oligomers join together, it called as polymer. Okay, so once this polymer become a macromolecule, once this polymer and become a macromolecule, and that macromolecule will have different characters like carbohydrates. And carbohydrate molecules like all sugars join together, single unit, simple sugars join together to become polymer, and then that polymer we call as macromolecule related to carbohydrates. Same way lipids. Okay, fatty acids, all fatty acids, different types of fatty acids join together, and that fatty acids we call as macromolecule or fats. Okay, like that. Proteins also, amino acids joins together and form as a polymer or macromolecule, and that we call as proteins. Okay, so vice versa. So here you can see different types of macromolecules we study in biochemistry. Okay, such as their structure and their physical characters and biological function of these macromolecules. So, including these macromolecules, the proteins, they have different role in proteins. We are going to see what roles they play inside the cell. And carbohydrates, different macromolecular carbohydrates are there, lipids and nucleic acids. Okay, so you can study these, macromolecules by studying enzymatic action and chemical regulation of metabolism and the molecular basis of genetic expression. And you can see different roles in vitamin synthesis, vitamin formation, and how these vitamins play compound what we call the prosthetic compounds in many macromolecules. Okay, so you can see many types of reactions, biological bio-oxidation reactions, and how energy will generate in these redox reactions, and how this energy is utilized in the cell for their regular metabolic activity, and how the cell save the energy in the form of energy rich compounds for further use. Okay, in later use, later metabolic activities. Okay, so as I said, do we have four macromolecules? Four macromolecules. In those four macromolecules, we are going to concentrate on proteins now. Okay, in essence, proteins are chains of different types of amino acids. So here you can see amino acid structure, amino acid having carboxyl group and amino group, hydrogen and R. Okay, R may be a simple hydrogen molecule or different complex molecule. Okay, depend on the R character, R size, R molecule, the amino acid character will change. Say for example, maybe either polar or non-polar or acidic or non-acidic, all like that. Okay, hydrophobic or hydrophilic, like that character will depend on this R, which link in the amino acid chain. So the carboxyl group, next carbon we call as alpha carbon, and then you can count from there how many amino acids are there. If more than 25 amino acids are there, up to 25 amino acid we can call as small oligomer, and if it is more than 70, 100 amino acids, we can call as peptide. Okay, if more than that, we can call as a polymer or macromolecule. Okay, so you can see the amino acid structure. We have different amino acids. Basically, you can see naturally occurring amino acids, 20 amino acids. You can see here. You can see here in this biological system. Okay.
Sir, we can see your first slide only.
Oh, it's moving. Sorry, is it moving now?
No, sir.
It's not moving.
Yes, sir.
One second, I will again. Stop presenting. Is it moving now?
Yes, sir. Now it's okay.
Yes, sir.
Now it is moving, isn't it? One second. I will push more. Let me know whether it is moving or not. Now I take the presentation mode. Thanks. Is it moving now? Slides are moving now.
No, sir. No, sir.
Can you see next slide? No, no.
No, sir.
One sec. Can I go to HOD office and present there? Because my system, since today it is giving some problem. Okay, I'm stopping here. I will move to HOD office. From there, I will present. Okay,
Okay, sir.
you can be there in this link. I will join there from that system.
Okay, sir.
Is it visible?
Yes, sir.
Hello.
Yes, sir.
Hello.
Yes, sir.
Slide is visible now.
Yes, sir.
Yes,
Is
sir.
it moving? Is it moving?
Yes, sir.
Yes,
Yes,
sir.
sir.
One sec. Let me put in slide presentation mode. How about now?
Yes, sir. It's moving.
Slides are moving, isn't it?
Yes, sir.
Okay, right. Thank you. Sorry for that problem. Okay, so as I said, biomolecule, four types of biomolecules, and you can see carbohydrates, lipids, and proteins and nucleic acids, and they have their importance in biological system, as you know, and different role they have in the system for their regular metabolic activities. And we are going to focus a little bit on proteins. So when you see proteins, and these proteins are polymers of amino acids. They are joined by different types of amino acids, and different types of amino acids we have. Basically, you can see naturally occurring amino acid 20 different amino acids you can see here. And then different amino acids naturally occurring or amino acids are, L configuration, and very little few amino acids are D configuration amino acids are there. So here you can see different types of amino acids depend on our nature. So if you see R just H, it is become to glycine, methane, methyl CH3, and CH. If chain is increasing, the complexity of that amino acid will increase, and the amino acid also change on this nature of this what R is in the amino acids. Okay, so you can see naturally occurring amino acids are generally R series, L series, and as I said, in very few amino acids in system, naturally occurring amino acids have D configuration. Okay. So depending on the H, character R in amino acids, we can have different types of amino acids. Non-polar amino acids like hydrophobic acids. 9 amino acids are there in this category, and uncharged polar amino acids. 6 amino acids there in this category. Charged polar and 5 amino acids are there in this category. So these are all very basic things. You can see the structure of amino acids and different types of amino acids, chemical nature, and all those things. Chemical composition, all those things you can see in any standards textbook. Okay, in any standard biochemistry textbook, biology books. Okay, in animals, proteins and polypeptides are polymer sequence of these 20 amino acids. Okay, so you can see the composition, one protein structure. These 20 amino acids, different positions, different difference in their number, difference in their composition. You can see in any given amino protein structure. Okay, so these amino acids basically given the information are encoded by the DNA DNA machinery. As we see in central dogma, DNA provides the information to synthesize RNA, messenger RNA will take the information to give a genetic code and give the genetic code to synthesize, to mix, to combine that amino acid, to join that amino acid with the growing chain. Okay, in plants there are few unusual amino acids that are sometimes found. As I said, D series amino acids, basically more D series amino acids in nature found in plants. Okay, and there are also other amino acids that involved in metabolism but not encoded by this DNA. Okay, so when you see D amino acids or L amino acids are encoded by genetic DNA, whereas amino acids are present those are not encoded by genetic code, those are not encoded by DNA, and these amino acids form during the metabolism. Okay, during the metabolism of these amino acids, byproducts they form inside the cell. For example, if you take ornithine and hydroxyproline and proglutamic acid, all these things are amino acid in nature, but they are not encoded by DNA. They are formed via metabolism. Okay, inside the cell. So as we see, almost all amino acids in nature are almost all amino acids in nature are alpha amino acids. Okay, so some type of amino acids like beta and gamma amino acids also you can see inside the cell, like gamma aminobutyric acid and nicotinic acid, anthralinic acid, all these related to beta and gamma amino acids. You can see the nature of the amino acid. Okay, the amino acid have different types of nature. Basically, we can call as amino acid zwitterion. Okay, so here the amino group sometimes get aminated and sometimes get positive charge, or carboxyl group get negative charge. Okay, depending on this pH or buffer change, this amino acid can able to act as a buffer. Okay, inside the cell. Okay, so they are zwitterion molecules, both positive and negative charge on their surface, on their structure surface, and depending on the situation, depending on the buffering capacity, environment, they can either become positive or negative by losing the proton. So because of their ionic nature, they have extremely high melting temperatures. So here you can see proteins have different temperature effect. Okay, say if you see some proteins some kind of proteins, reverse transcriptase enzyme. Okay, reverse transcriptase is enzyme. It is enzyme in nature, but it is a protein. Okay, so that protein is heat sensitive. If you incubate that protein at 42 degrees for more than 15 minutes, it will get denatured. Okay, it will lose its activity. Okay, so another protein, good example, if you take tag DNA polymerase. Okay, tag DNA polymerase is an enzyme, is also a protein. So you can incubate that protein at up to 100 degrees, more than 100 degrees also you can incubate. So because of this ionic strength, these temperature, melting temperature are, extreme. They have extremely melting temperature because of their ionic nature in the sample. Okay, so here you can see amino acid and another amino acid join together by losing a water molecule and form as a peptide. So you can see a peptide, red bond is a peptide bond here. So by this action, by this way, they become dimer, they become trimer, and they become oligomer, and they become peptide or polymers. Okay, so peptides are linearly polymers and ranges from 8 to 4,000 amino acid residues. Okay, if more amino acids are there, the complexity of that protein polymer will increase. Okay, complexity of that polymer and chemical nature of complexity of chemical nature and function and all those increase, and molecular weight also increase as well. Okay, depending on increasing the amino acids, the protein molecular weight increases. So here you can see how many amino acids, different combinations. We have 20 amino acids, different various combination. You can get different length of the amino acids. Total number of atoms in universe estimated as 9 to 10 to the power of 78. Okay, so if you take acid and second amino acid, you can get 400 different molecules. If you take 3 amino acid, 20 to 28,000 different molecules, we have 20 amino acids. So you can imagine how many different types of proteins are present. Okay, when you see the genomics and proteomics, genomics is very easy to to understand the gene structure, to understand how many genes are present. When you see the complete human genome, only 30% of genome is expressing. Okay, roughly estimated it is expressing into state. Okay, remaining 70% is junk or whatever repeating sequences, whatever you call. Okay, so 30,000 genes are producing from the complete genome sequence. So that means the human beings require 25 to 30,000 genes only. Okay, so it is calculated. So when you see the proteomics, when you see this proteomics composition of these amino acids and complexity of these amino acids, complexity of these proteins, you can understand how many variations, how many variant amino acids are present in a protein. Okay, so that is because of we have 20 different amino acids composition. Because of 20 amino acids, they have a chance to have different combinations. So because of these different combinations of these amino acids, they have different characters and they have different chemico-physiological activities and they have different molecular weight in nature. Okay, that is the complexity of that proteomics. Okay, so these proteins, these amino acids can be called by their names. Okay, as we see alanine, and by their structure or their 3-letter code or 1-letter code. For example, if you see tyrosine is its chemical name, and its structure here you can see. If you are very familiar with the amino acid structure, if I give this structure, it is very easy to you to tell this is tyrosine. Okay, so we can use 3-letter code TYR, capital TYR, or we can use a 1 code letter, genetic code letter, capital Y. Okay, so this is a aromatic having hydroxyl group. Aromatic ring is there and hydroxyl group is also there in this amino. Okay, by this way all amino acids are called by their name, structure, 3-letter code or 1-letter code. So here you can see different types of amino acid by their name, 3-letter code and 1-letter code. So their structure, and you can see their PKP, PK1 and PK2 values and average occurrence in a protein. So occurrence of the proteins also calculated. So for example, leucine, you can see in this table, leucine is the highest occurrence in any proteins. Okay. So like this, you can see different types of amino acids. Their structure is present inside the cell. Inside. Okay, present. Sorry. Okay, next proteins. These proteins, what they have role. When you see proteomics, it is a big complex study. So when you see their role, they have different roles here. All in all biological system, proteins are the main having main functional activity. Okay, so for example, if you take few, some they are involved in structural characterization of a cell. Okay, structure. Okay, so actin and myosin are good example, those present in the structural structure of that particular cell. Okay, and then if you see a few portion of the proteins belongs to a protection thing, those are called as immune proteins. For example, if you take antibodies are good example for the immune system. Not only antibodies, different types of proteins are present inside the system. They have their protection role, like complementary proteins, complementary complex proteins also belongs to proteins. Okay, these antibodies different experimental purpose to understand whether the foreign protein is present or any antigen is present or not by ELISA test. Okay, and then most of the protein category belongs to is a special category. Those are called as enzymes. Okay, these molecules recognize a specific reactant molecule that we call as substrate and catalyze the reaction between them. Okay, so they recognize the specific molecules. Okay, those we called as example. A is one molecule, B is another molecule. This enzyme will react with these two molecules and may interact with each other and give a product. Okay, C product. Okay, so this engine, what way, how it is helping by lowering the reaction activation energy. Okay, so for example, you can see this one. The engine speed up the reaction by rate of 10 to the power of 10 or more. A reaction that would normally takes over 3,000 years to completely might take less than a second if you add an enzyme in that reaction. Okay, you can see the specificity of that particular enzyme, particular reaction. The reaction will take 3,000 years to complete. If you add an enzyme here in this type of reaction, that reaction will complete within seconds. Okay, so that's the way the energy it reduces. So any reaction, if you take any reaction to start, we need to provide some kind of energy by increasing the heat and providing some kind of motivation energy to start that reaction. Okay, here these enzymes involved in this catalysis or in this reaction by making interact with each other and lowering the energy. Okay, so basically it will make the interaction between two molecules and interact by lowering the energy and make the substrate from the two reactions without consuming this molecule, this enzyme. Okay, so enzyme is not used up in this process and is free to catalyze same reaction with new set of substrate. So once A and B interacted and become C, so enzyme will be there. So A will not be there in this reaction. Okay, you can see only C. Okay, so enzyme will be there. When you add new A and B, again it is ready to interact with that. Okay, in this process, the enzyme is not consumed. It is not utilized. That is the meaning here. Using various modifications, the activity enzyme can be regulated. Like genetic you can see coming classes. You can see different modifications how we can make enzyme to act more or activity or to stop the activity. Okay, so we have different mechanism. These mechanism can be utilized to control the enzymatic activity, because it is very important to control the enzymatic activity. Otherwise, if this enzyme is keep on acting on the substrates, all molecules will become substrate molecule will become product molecules. Okay, then cell will look for substrate molecule. Cell will have if there is no substrate molecule, these active enzyme may have different side effects interacting with other molecules and problem. Okay, good example I will tell you, arthritis. Okay, in this arthritis, the problem is the enzyme which is involved in this conversion of this substrate molecule into product. This enzyme is keep on active. So unless we provide some inhibitory molecule, this enzyme will not get inhibited. So it will cause the problem. That's the reason we get some kind of joint pains and arthritis pain in the arthritis disease. Okay, so here our system will have molecules that enhance the enzymatic activity, and I will have molecules that reduce the enzymatic activity. So that we call as regulation. Okay. So now coming to the point. So enzymes. What is enzyme? What is enzyme definition? Enzymes are biological catalysts, are chemicals that speed up the rate of the reaction between substrates without themselves being consumed in this reaction. Okay, this we can give as a definition for this enzyme or enzymes. So enzyme names, okay, there are different types of names were used. Okay, so go back to 1950s, 1960s, 1970s. Many research groups were started working on these enzymes. Okay, many groups were working and many groups were identifying the new enzymes and giving the names. Okay, different names are we can see. So different names we can use. Tribal names. Okay, gives no idea on source and function or reaction catalyzes by that enzyme. So tribal names will tell you nothing. Okay, but that name indicates the enzyme. Okay, so for example, trypsin. Trypsin is housing thrombin, pepsin. These are all the enzyme in nature. Okay, so when you study these enzymes, then only you will come to know what is its activity, what is its role, what is its function. Okay, so this type of names we can call as tribal names, and then systematic name came up. Okay, so people you got confused by these tribal names. They have to read this. They have to study that function of this enzyme, what is that molecule name is, what is that enzyme is, what is that molecule is. Okay, so later to avoid this confusion, people started giving some kind of special nomenclature to these enzymes by systematic way. Okay, according to the International Union of Biochemistry, an enzyme have two parts. Enzyme name has two parts. Okay, so in 1970s, all enzymatic research groups are joined together. Okay, somewhere in Geneva, and then they discussed about this enzyme nomenclature. We have to give specific names to this enzyme, otherwise we will get confused. Okay, so for example, one guy who is working in India, he's working on one enzyme. He will claim that I discovered this enzyme. Same enzyme was somewhere in US or somewhere in South Africa. They are characterized an enzyme and they given their own name. So when they are joined together, they call this name as this name, their name X, and this guy will call that other name. So when it comes to literature, both enzymes are same, but the names are different. Like this in literature, when you see 1,000 names are different, but they have same function. Okay, so to avoid this kind of confusion and differences, these research scientists join together and form a union, world union. Okay, so that is called as International Union of Biochemistry, and this union will look into the activity of that enzyme, character of that enzyme, physical chemical properties of that enzyme, and what reaction it will do, what is the substrate, and all those things, and naming. So here two things they followed. One is it will have the substrate, okay, first part, and second part will have ase. They suffix ase to that first part. So substrate will have ase. Okay. So for example, if you take enzyme lactate, lactate is a substrate. Okay, dehydrogenase. So ase is there. What it is doing? Dehydrogenation. So it is removing the hydrogen molecule. So lactate dehydrogenase. So you can see the enzyme which is working on, understand that molecule is an enzyme. It is working on lactate. Okay, once you go into deep, how it is working, what kind of work it is doing on this substrate, you can understand later. Okay, so by this way you can see the first part is the substrate on which it is working and suffixing the age. Okay, what type of reaction. Okay, second part is the reaction catalyzed by the enzyme. This part ends with the suffix age. So second part, the catalyzation of the reaction, and this catalyzation of reaction, what role it is working, and suffix with ase molecule. Ase. So you will get substrate and ase, that is called as, for example, lactate dehydrogenase. So as I said, this group, International Commission, again, they simplified this nomenclature by giving the enzyme commission number to this classification. Okay, so enzyme commission number is strictly speaking, enzyme commission number does not specify enzyme but enzyme catalyzed reaction. Okay, it differs if different enzymes catalyze the same reaction, they can then they receive the same catalyzer EC number. Okay, so this is about how where they formed and what they have done, all these things. Okay, so in this enzyme commission number, you can see numerical numbers separated with the dot, separated with the full stop. Okay, so for example, EC 3.4.11.4. 4 numerical numbers will be there, and these numbers are separated with periods or dots. Okay, so if you see EC3 is hydrolyzed enzyme. Okay, and enzyme that uses the water to break some molecule bond. Okay, and EC.4 are hydrolysis that acts on peptide bond. Okay, EC 3.4.11, these hydrolysis that cleave off amino acid terminal from the polypeptide. So it works from the amino acid direction, amino acid terminal from amino acid side, terminal side. Okay, and EC 3.4.11.4 are those cleave off amino terminal from the tripeptide. So this enzyme cleaves the bond, peptide bond, okay, from the amino acid terminal, and then last digit indicates the 3 amino acid, third peptide bond will be cleaved. Okay, so here you can see the first one is class of the enzyme, and subclass and subclasses are divided by different numerical numbers pointing with dots. Okay, so here you can see EC1 enzymes belongs to oxidoreductases, EC belongs to transferases, EC3 belongs to hydrolysis, EC4 ligases, EC5 isomerases, EC6 ligases. Okay, so like this they separated into they classified into 6 categories of these enzymes, and then all these enzymes based on the substrate, based on the reaction, they grouped them in these 6 categories. Okay, for example, reduction reactions, all oxidation and reduction reactions are placed under this group. Okay, all type of oxidation which remove the electrons, which accept the electrons, remove the protons or accept the protons, all this type of oxido-reduced reactions, reduced and oxidation reactions are placed in this group. That is called as oxidoreductases. Okay, and second one is transferases. In this reaction, the functional groups or molecules are transferred from one substrate to another substrate. Okay, so this type of reactions they grouped in this second type, transferases. Okay, so hydrolysis, in this reaction that they will break the bond water molecule. Okay, so those type of reactions are kept under this group. Ligases, non-hydrolytic reaction, removal of subgroups from substrates. So you can see without water molecule, those reactions break the bond or transfer the functional groups from one molecule to another molecule by breaking the bond. This type of reactions they kept under this group, fourth group, ligases. So where the bond is cleaved in absence of water molecule. Okay, so the functional groups are transferred from one group to another, one substrate by breaking the bond without water molecule. So those groups all kept under this ligase category. Okay, so you can see the examples also in the enzymes. So isomerases, here these molecules, these enzymes will transfer, interchange of sub molecule from one carbon to another carbon. Okay, interchange of their double bonds. Okay, all these molecules, all these groups of all these enzymes kept under this group. Okay, isomerases, mutages, enzymes transfer this hydroxyl group from one carbon to another carbon within that substrate molecule. Okay, like these reactions. Okay, and then ligases are molecules, enzymes. They make the new bond. Okay, so they synthesize the new bond. So you can see different types of enzymes present here. Synthetases, all belongs to these molecules where they make new group. So in this process, they require energy. So in this chemical reactions, ATP or GTP or energy-rich molecules will be utilized to provide the energy to synthesize new bond. So they entire enzymes are classified, categorized in these six classes of molecules. Yes or no?
Sir, sorry to interrupt you, but
No
actually we
problem.
have a class at 10:30.
Oh, okay then, no problem. See, my problem is me. So will if I have will join by 9:35:40. Okay, here
Okay,
are
sir.
okay,
Okay, sir.
right.
Okay, sir.
Okay, see you next class.
Thank you, sir.
Thank you.
Thank you, sir.
Thank you.